Hello everyone, I'm Pratibha Sharma. Today I'm going to discuss chapter number 2 of class 12th. Under that we will be studying two topics. One is megasporogenesis and the second one is development of female gametophyte. In my earlier classes, I taught you what is microsporogenesis, how microspores are formed, and later on how they develop into male gametophyte, which produces male gamete. So today we are going to study how megaspores are formed and how these megaspore changes into female gametophyte, which contains the female gamete. So pistil, as we already know, pistil belongs to the floral hole gynoecium. It is an essential floral hole and is representing female reproductive part. In a flower, we find that there may be a single pistil present. In that condition, we call the gynoecium as monocarpellary. But if there are more than one carpels present in a flower, then it is known as multicarpellary. Now, if the carpels are fused, all the carpels, they are fused, then that condition is known as syncarpous. Like in this particular case, you can see pistil is one, but it is formed by the fusion of three carpels. How we came to know about this? Because here you can see that there are three stigmas. And when we take the section of the ovary, inside the ovary, there are present three chambers, which you can call as locules. Okay, so this is multicarpellary syncarpous ovary. Now, the multiple carpels, they may be free, like in this particular case where there are three carpels and all their parts are separate, they are not fused. And each one is having a separate ovary, and inside the ovary there is only one chamber, which indicates that it is a single carpel. Okay, so if the carpels are free, then that condition is known as apocarpus. So, pistils in a flower, there may be one pistil, then monocarpellary, more than one pistil, multicarpellary. If the carpels are free, apocarpus, and if they are fused, syncarpus. A pistil is having three parts. First, at the top is present stigma. It receives the pollen grain and pollen grain germinates over the stigma. Then there is a long tube and that tube is known as style. Through the style, pollen tube, it travels downward towards the ovary. Ovary is the basal swollen part of the pistil. Inside the ovary, there are present locules, chambers, and inside these chambers, ovules are present, which are attached to the wall of the ovary by means of placenta. In an ovule or in a locule, you will find that there may be one or more ovule present. Next is regarding Megasporangium. Okay, the other name for Megasporangium is ovule. Since the formation of megaspore is taking place inside the ovule, that's why the entire ovule is also known as Megasporangium. This figure is the diagram of anatropous ovule. In your exams, you will find that many times they specifically ask you draw a well-labeled diagram of anatropous ovule. So, you have to draw this diagram. Okay. Now, an ovule consists of two parts. The central mass of diploid cell is known as nucellus, and covering the nucellus there are two envelopes known as integument. Okay, so an ovule or megasporangium is having two parts. One is nucellus and the outer to the nucellus is present integument. Now this ovule, since it is present inside the ovary, it is attached to the wall of ovary by means of a short stalk, and that stalk is known as funicle. The point or the junction where the funicle is attached to the body of the ovule, it is known as, here it is attached, so this portion is known as hilum. Okay, now you can see in the figure that the integument doesn't cover the nucellus completely. It leaves a small opening at one of the end, and that is known as micropyle. In majority of angiosperms, the pollen tube enters inside the ovum through this micropyle. Opposite to the micropyle end, the basal part of the ovule which is present is known as chalaza. In the nucellus, the development or formation of megaspore occurs, which later on develops into female gametophyte, which is known as embryo sac. Okay, so this is about the description of anatropous ovule. You should know the function of each part. What is nucellus? What are integuments? Okay. Now, how in the nucellus the megaspores are formed? So the process of formation of megaspore is known as megasporogenesis. Megaspores, they are formed from the megaspore mother cell. So this diagram is given in your textbook. You have to draw exactly the same type of diagram. Here you can see these are the cells of nucellus. This is the micropyle end, the end where a small opening is present. Okay, so towards the micropyle end, in nucellus there are many cells present. Only one cell, it acts as megaspore mother cell. This megaspore mother cell, this is the nucleus of megaspore mother cell, which is diploid. Since angiosperms are diploid, they are sporophyte and sporophytes are diploid. So megaspore mother cell, it undergoes meiosis. Okay, one of the megaspore, one of the nucellar cells in the micropylar region, it differentiates and forms the megaspore mother cell. It divides by meiosis. After meiosis 1, it results in the formation of megaspore dyad. That is, 2 megaspores are formed. These 2 megaspores are haploid, which further divides by meiosis 2 and results in the formation of tetrad of megaspore. So we get 4 haploid megaspores. Okay, now out of the 4 megaspore, the upper 3, these 3, they degenerates and only one survives, which enlarges in size and is known as functional megaspore. So in megasporogenesis, from one megaspore mother cell, we are getting only one megaspore. So this entire process is known as megasporogenesis. And this functional megaspore, later it develops into female gametophyte or embryo sac. So at the end of megasporogenesis, we obtain one megaspore. That megaspore then develops into female gametophyte. Okay, so first of all, we find that one single functional megaspore, okay, which is there, it is going to develop into the female gametophyte. Now, since from one megaspore, one embryo sac or female gametophyte is formed, this type of development is known as monosporic development. the nucleus of the functional megaspore, it divides by mitosis and it results in the formation of two nuclei. Okay. So here you can see this is a two-nucleate embryo sac. Okay, initially in the microspore there was only one nucleus. It divided by mitosis. So two nuclei are formed. The two nuclei, they move to the opposite pole. After that, these nuclei, they further divide successively two times by mitosis. Okay, this division is free nuclear mitotic division. The cytoplasm doesn't divide. So after second mitotic division, at the two poles, we get four nucleated embryo sac. Then it further divides by mitosis and that results in the formation of 8 nucleated stage of embryo sac. Okay. So it results in the formation of 8 nucleated embryo sac. Now after the 8 nucleate stage, the cell walls are laid down around them. Okay. So out of the, you can see here, the four nuclei are there on the opposite pole. One nuclei from each pole, it migrates and come to lie in the center. So at each pole there are left 3 nuclei. Now around these nuclei, the cell wall is laid down or formation of cell wall occurs. So that results in the formation of 3 cells on each pole and the 2 nuclei which are present in the center, around them also cell wall is formed. Okay, a common cell wall is formed. So here you can see this is the micropylar end. Okay, so at the micropyle end, the 3 cells which are formed. In the center, the cell which is formed is known as egg cell. Okay, and lateral to the egg cell, there are present 2 cells known as synergids. Synergids are having finger-like extensions in them. These structures are known as filiform apparatus. When the pollen tube enters inside in one of the synergid, the filiform apparatus guides the pollen tube towards the egg cell. This whole structure present at the micropylar end is known as egg apparatus. So an egg apparatus consists of 2 synergids and in the center female gamete, that is egg cell. Now at the chalazal end, the 3 cells which are formed, they are known as antipodals. The 2 nuclei which were there in the center, which are known as polar nuclei. The cell in which they are present, that cell is known as central cell. This is the largest cell of the embryo sac. So here this complete structure is the mature embryo sac. Okay, so an embryo sac, it consists of 7 cells and 8 nuclei. Okay, how? So you can see 3 antipodals and 2 synergid, 1 egg cell. So 6, and in the center there is central cell. So it is 7-celled 8 nucleate structure. Okay. So 8 nuclei because in the center, in the central cell, there are 2 nuclei present. Okay, in exam, you might get question like draw the diagram of mature embryo sac or 7-celled 8 nucleate structure of embryo sac. So this is the structure of embryo sac which is formed before fertilization. It is called embryo sac because when an egg will fuse with the male gamete, it will form zygote and that zygote later develops into embryo. So as this structure is going to contain embryo, it is known as embryo sac. And since it contains female gamete, that is egg, that's why it is also known as female gametophyte. Okay, that is all about megasporogenesis and development of female gametophyte or embryo sac. Now some questions which are asked in board exams based on these two topics. So as I told you, uh, you might get diagrams. Okay, so label diagram of anatropous ovule, or you might be asked draw a labeled diagram of mature embryo sac. Okay, and then like how many haploid cells are present in mature female gametophyte of a flowering plant? So they have asked about cells. So there are total 7 cells and you have to name them. So 3 antipodals, in egg apparatus 2 synergids, 1 egg cell, and in the center there is central cell. I hope you must have enjoyed this lesson, and if you like my video, please give a thumbs up and do subscribe my channel so that you will get notification of the videos which I upload. Thank you.